Hi guys, I wanted to add a little detail today on some of the regulation of the cell cycle and how it's controlled. So there's a couple, we mentioned that these checkpoints existed, but we hadn't really talked about what they were or how they work. So I want to do that today. And one of the players here is a kind of a protein called cyclin. And these are proteins that were named this because if you looked at their levels over time, they would go up and down and up and down. And then this timing was mapped onto the cell cycle itself, and it was noted that the highest level of, for example, the first cyclin we'll talk about was highest at a specific phase in the cell cycle. All right, so we had two checkpoints that we talked about. There's actually another one that's in mitosis. The first checkpoint that we talked about is this transition from G1 to S, okay? And during this checkpoint, the cell is sort of asking itself questions, okay? The cell is asking itself questions like, do I have a sufficient cell size? Is there enough nutrients available? Am I getting the right signals from other cells? Okay. This protein, this G1 cyclin, works together with another protein called the cyclin-dependent kinase, or CDK. And when these two are together, when the G1 cyclin is together with the cyclin-dependent kinase, this complex can activate the proteins necessary to go from G1 on into S. Okay. Remember, kinases are proteins that transfer phosphate groups. Okay, so they're taking a phosphate from ATP and transferring it onto a protein needed to initiate S phase. Okay, so this cyclin kinase complex is what's active. This kinase by itself will not act to phosphorylate the targets. So when you have that complex, you can then, if you're answering the right answers to these questions, you can proceed from G1, or your gap phase, on into the S phase. All right? And as this graphic shows here, it's really the cyclin with the CDK, the cyclin-dependent kinase, that is active. Okay, and then this can transfer phosphates onto the target protein, and this target protein phosphorylated version is now active and will initiate the progress into S phase. Okay, the S phase is obviously where the DNA is replicated. The second checkpoint so there's no checkpoint between S and G2. The second checkpoint is located between G2 and M phase, or mitosis. All right. So here we have a different cyclin. So this is a different protein. All right. We have our M phase cyclin. And when this interacts with its cyclin-dependent kinase, if you have these two things together, you have a mitosis promoting factor. Also known as the MPF. Okay? And this factor must be present in order to progress into mitosis. Okay? So cell size is adequate if your chromosome replication has been successful, and if you form this complex, you can progress from G2 phase into mitosis. Okay, so those are the two checkpoints. Okay, the second one, you form your mitosis promoting factor, your M phase cyclin, your cyclin-dependent kinase, and again, that can initiate the series of events leading to mitosis. All right, the final checkpoint is in metaphase. Okay, and this question being asked here is, are all chromosomes attached to mitotic spindles? Okay, if they're not, right, you could have improper segregation of chromosomes. You could get a case where you did not have separation of sister chromatids. Okay, this could become a problem, right, if we're talking about misallocation or breaking of chromosomes. If this is not the case, you could get daughter cells with an incorrect complement of DNA. Okay, so there's a cyclin that is at work here. Important to note that these cyclin-dependent kinases these cyclins are themselves degraded, right? So the initial plot that I kind of showed you where you had high levels and low levels. So after these cyclins are formed and they promote, they together with their partner proteins are activating progress through a checkpoint. The cyclins themselves are degraded. Okay, and this production of cyclin happens during the growth phases. Okay, so this is one of the ways the cell is saying, I am big enough because I've been able to synthesize enough cyclin. Okay, so these are three checkpoints that are part of the cell cycle. They are important, right, in maintaining the proper level, proper pace of growth. Obviously, if these go wrong, they can start the progression towards cancer. So, obviously, the cell cycle is related very closely to the process of development of cancer. So if cancer is uncontrolled or unrestrained growth, there's kind of a couple of ways that you can get there, okay? If we look at the last panel of this, either you can have an overexpression of a protein that stimulates the cell cycle, or you can lose function of a protein that normally inhibits or regulates the cell cycle, okay? There's a little bit of terminology you need to know, and that is the term oncogene. Onco means cancer. So an oncogene is a gene that, this is a protein product, right? A gene encoding a protein that promotes the development of cancer. Okay, and very often we can consider this as a mutated form of a natural protein, okay? So the form of this, the proto-oncogene, this is a gene that has a normal function, in growth, all right? This is a protein that has a normal function. Once it's mutated, it becomes an oncogene, okay? All right, so let me just go through a quick example of this. One example is the protein RAS. So RAS is a G protein, so it responds to a signal. This is a receptor that is dimerizing upon binding. It's phosphorylating itself, starting the process of releasing RAS through a series of, for example, probably kinases. A transcription factor is activated, and that causes gene expression. Okay, if you have a mutated version of RAS, it is hyperactive, so it's sending much more signal here than the wild type RAS or the unmutated RAS. Okay, so you get a much bigger expression of your stimulatory protein. Okay, so this would again cause an increase in cell division, progression towards a cancer. Another very famous example is P53. So this is a protein that normally acts to promote the production of a protein that normally inhibits the cell cycle. So P53 is a transcription factor. That means it sits on DNA, helps recruit the machinery that turns it into RNA, which eventually leads to protein. All right, but if you have a mutated form, this form, for example, let's say the mutation came from UV light, this form could not bind to the DNA, and you will not be producing this inhibitory signal. Okay, P53 is mutated in something like a third of all cancers that have a familial link to them, so it is an important name to know. All right, so that's kind of two ways you can get to increase cell division on the pathway to getting cancer. All right, hope that helps.